Good morning, good afternoon, and welcome to the 9th in our Webinar Wednesday series. Shalom, bonjour, and hello, and I suppose because of Debbie, we should say hola to all of you who have registered from around the globe. To be exact, we have people from Israel, England, and the United States, and of course, Canada. I am Mark Mendelson, the Chief Executive Officer of the Canadian Associates of Ben-Gurion University, and it is once again my pleasure and honor to be moderating today's program. I am more than thrilled to tell you all that we've broken all previous Webinar Wednesday records by having well over 150 participants register for today's call. This just shows how urgent and relevant today's topic is and how affected so many people in our community are by family members with dementia or Alzheimer's. And we have two of the most brilliant and award-winning of the younger researchers at Ben-Gurion University, Drs. Debbie Toiber and Dr. Tammy Riklin Raviv. I must say that I spent 3 to 4 days with Debbie in Vancouver and Winnipeg, who spoke at events there, and she is definitely a rock star. And I'm sure Tammy is as well, being the mother of 4 kids. Sorry, I'm embarrassing you both, but that's just the way I roll. On behalf of the lay leadership of CABGU, Canadian Associates of Ben-Gurion University of the Negev, aka CABGU, I welcome you and thank you all for joining. Please note that we are streaming live on Facebook. I would also like to warmly welcome our participants as well as our BGU colleagues from US and Israel. Once again, the format of today's webinar is this. I will first introduce Dr. Tammy Riklin Raviv, who will talk for about 12 to 15 minutes. And then I will introduce Dr. Debbie Toiber. I will give each presenter a 2-minute warning before their time is up. And then there will be a Q&A segment. And I must tell you that we are already inundated with questions. As always, Montreal Executive Director Simon Ben-Simon will be handling the question and answer period. And Zach Ostrov, Executive Director of Winnipeg, our in-house tech expert, will be handling our technical issues. Hopefully, we won't have any. Comments or questions, you will see the icon at the bottom of your screen, and you'll just have to click on it to submit your comments or questions. The topic of today's webinar, Understanding Alzheimer's and the Aging Brain. Neuroscience is one of the first scientific disciplines to recognize the need for interdisciplinary collaborations to solve complex problems, and many of the field's achievements are the result of the collaborative work of the researchers from different disciplinary backgrounds. In many of the highest ranking universities, this interdisciplinary approach has been translated into interdepartmental neuroscience research and training programs. With this in mind, BGU has established the Interfaculty School for Brain Sciences with the participation of the faculties of Humanities and Social Sciences, Engineering, Natural Sciences, and Health Sciences. This is the most unusual approach and not only in Israel, but throughout the world as well. And at this point, it is my honor to introduce our academic participants, Drs. Riklin, Raviv, and Toiber. Tammy Riklin Raviv is a senior lecturer at the School of Electrical and Computer Engineering of Ben-Gurion University of the Negev. She joined BGU in 2012 and established the group of biomedical image computing. Her main research interests are computer vision, machine learning, and biomedical image analysis. She's an associate editor at the IEEE Transactions on Medical Imaging, on Medical Imaging Journal, and a TC member at the IEEE Bioimaging and Signal Processing Committee. She holds a BSc in Physics, MSc Computer Science, magna cum laude from the Hebrew U, and we will not hold that against her. She received her PhD from the School of Electrical Engineering at Tel Aviv University. And like Debbie, during 2010 and 12, she was a research fellow at Harvard Medical School and the Broad Institute of MIT and Harvard. Prior to this, she was a postdoctorate associate in CSAIL, MIT. Tammy, the computer and the screen is all yours.
Hello, good afternoon. I'm very happy to be here and thank you for the introduction. In my talk today, I will explain how we can exploit big data and deep learning in order to understand brain aging and to do age predictions. Now, the basis of the talk today is a recent paper we published in the Human Brain Mapping. It is a collaboration with Professor Galia Avidan of Cognitive Neuroscience, her student Gidon Levakov and Gidon Rosenthal, and Professor Ilan Shelef. Ilan Shelef is a radiologist, is the head of the Radiology Association in Israel, and he is at the Soroka Medical University. So you can see it's a collaborative effort, neuroscience, electrical engineering, and radiology. Now, as you may all know, our chronological age is associated with brain anatomy. So processes such as neurogenesis, cell migration, synaptic growth, and synaptic pruning takes place when we are young, while processes such as brain atrophy, neurodegenerative processes, inflammation, vascular changes may take place when we grow older or when we suffer for some neurological disease. Now, if you look at the literature, and here is just a small subset of the numerous papers exist, you may find out that there are a lot of work that trying to associate brain age and brain anatomy. So we all know that brain undergoes complex structural and changes throughout aging. These changes are not uniformly distributed across the brain. They are accompanied by cognitive decline and memory deficits, yet more extensive processes are evident in neurological diseases such as Alzheimer, and here I refer to the next talk. Now, the goals of this study is first to do prediction, that is to develop a method to identify brain maturation independently of biological age. And at the same time, we would like to do inference, that is to develop a method for localizing these brain regions that are responsible to the morphological changes related to aging. So when we talk about prediction, we must consider the main tool we are doing the prediction, which is deep neural networks. Now, as you may all know, artificial neural networks are inspired by biological neural network. And here you can see very nice drawing on your left of Ramon Cajal illustrating biological nervous system. While on the right-hand side, you will find out a schematic drawing of deep neural networks. Now, what we do is we take artificial neural networks inspired by our biological neural network in order to study our biological neural network, that is our brain. Here you can see side by side the biological neuron versus the artificial neuron. So if you look at the lower part of this slide, you will find out a schematic drawing. You can see some input artificial neurons. They are connected to another neuron by a synaptic connection, that is these WIs. These are somewhat analogous to the dendrite. All this information are summed, actually a weighted sum, and then there is a nonlinear activation function. This could be a threshold or a sigmoid, and in analogy to the neural, to the biological neuron, you can think of the axon as carrying out the output of the neuron. Now, actually, when you talk about biological neuron, the model is much more complicated, and we talk about action potential, but we don't need this here, so we move on. Here is again a schematic drawing of a deep neural network. It is not so deep, it's just a schematic drawing. Usually we have an input layer, an output layer, and many hidden layers. Now, if you consider a single neuron, okay, it is here, it is connected by an edge, which has a weight, Wij, that is, every neuron I may be connected or not connected to another neuron in a consecutive layer, and the connection is marked by Ij. When I train a neural network, what I do is I learn these weights. Once they learn, once the neural network learned, I fix these weights and then I do the prediction. I'm not going into details. You may be familiar with neural network that perform a classification task. That is, if we have a cat and a dog, the neural network is trained to identify whether the input is a cat or a dog. In the same manner, I can use prediction in order to solve a regression problem. And in this case, I may present in the input layer to the neural network an image of a brain and then predict the brain age. Specifically, we use convolutional neural network. Again, I will not get into too many technical details. All I can say is that convolutional neural networks are greatly inspired by the visual cortex, and they do an action that is similar to what is known to be convolution. In this work, we use an ensemble of convolutional neural networks. That is, we explore the power of using many, many learning machines. Each of these networks were trained to predict brain age. And then once we have new brain images, we use them in order to get the age. In order to do that, we use more than 10,000 images, T1 MRI, from different data sets. And here you can see the logo, but here, if you look at this table and you give a closer look, you can see specifically the source of these data sets, the number of patients, their average age and the standard deviation, as well as how many are females and males. Our measurements are the mean absolute error. That is, this is how we measure what is the error between the predicted brain age from the T1 MRI and the chronological age of the patient, and the Pearson correlation between the prediction and the true value. Now, I will get right away for the results. So these are our age prediction results, and you can see that the error is about 3 years. Now, considering that our age range, and here you can see the chronological age with respect to the predicted age, varies from about 4 to 19. You can see that this is really a nice prediction, and we are almost on this straight line. If you look at the boundaries of this plot, you can see the histogram of the ages. And it's easy to notice that here, on the ages about 30 to 50, we don't have as much as example as patient at the ages, till 40 and from 60 and on. This is the weakest part of our study, but we are working on that. Now, if you look specifically on each of the data sets that we use, you can see the prediction for each. And what is more important is the prediction uncertainty. So since we're using an ensemble of convolutional neural network, we can actually have the average prediction and the variance of the prediction of each of these networks. And what is nice to see is that the error in prediction correlates with uncertainty. Now, what should be the source of error? It could be that we thought that the subject we are investigating is healthy, but in fact, he has some neurological disease he or she is not aware of. So this should be taken into consideration, and I will get to this point later on. Now, what is more important is not the brain age per se, but what contributes to the brain age. Which regions in the brain actually make us think that this is the right prediction? How can we explore that? Maybe you heard that people say, well, neural network is like a black box. Okay, you train a neural network on a set, on a training set, and then do the prediction, and you actually don't know what's going on there. But there are some methods in order to investigate, and we are going to use them. So let's say you do a slight change in your input, and then look at your prediction. In other words, you actually take the derivative of the output with respect to the input. There you can find out what in the brain makes the prediction to be as it is. And this is what we did. So we took all our test subjects, project the explanation map that are generated by the derivative of the output with respect to the input. That is, we did a slight change in the input in order to find out what influenced the most on the output, merge them together, okay, after projecting into the anatomical space and doing registration into the MNI space, and then we can threshold. Once we threshold, what is highlighted here on the brain is simply those regions that contributed the most to our deep learning based edge prediction. Now, since we have an ensemble of networks, actually 10 networks, we can merge them all together and get the contributing regions in higher confidence and the uncertainty. So in order to demonstrate that, here you can see some slices of the brain, sagittal, accents, and so on. And you can see also the explanation score. So if you're red, these are thresholded, by the way. So here you can see exactly all these regions that contribute the most for the edge prediction. And here, after doing this study on our entire database, with the ensemble of neural network, we actually found out what region contributes mostly to aging in terms of extent, that is cluster size, and the peak of our score. What we found out, and the list is longer, you can see it all in the paper, is that the ventricle and the ambient cistern actually contribute mostly to aging, but there are other regions as well. Now, in order to find out if we're doing right or not, we compared our results with those obtained using voxel-based morphometry. So on the upper side of this panel, you can see our results versus voxel-based morphometry results, and you can see that they are highly correlated. So just to conclude.
Tammy, 2 minutes.
Yeah, thanks. I'm at the end. Just to conclude, individual chronological age can be predicted from T1 MRI of the brain. It is possible to localize brain anatomical regions that allow such prediction. And the key components are using big data. So we had brain scans of 10,176 subjects. And then we use deep learning, that is an ensemble of 3D CNNs. Main results at prediction accuracy with error of slightly more than 3 years. And we found out also that the ventricles and the cistern govern this prediction. Now, having this, what's next? So future direction. We would like to generate 4D explanation maps that varies with age group and gender. And then we want to generate a region-based prediction. This is what we are working on currently. Now, what is more interesting is associating and under an overestimation of brain age to lifestyle, as well as neurological, neurodegenerative, and neurodevelopmental diseases. Thank you.
Am I on, Simon? Yes. Tammy? Wow. When I was in science at McGill, I should have listened more intently. But obviously, incredible research and the demonstration of your of your brilliance in this subject was just amazing, I must say. Thank you so much. Next, Dr. Debbie Toiber, who you will know as soon as she speaks, was originally from Mexico City. She made Aliyah, as I did, except she did it as a teenager, and I was a little older. She obtained her doctorate from the Hebrew University of Jerusalem, where she also met her husband-to-be, himself a researcher, but at Hebrew U and not at BGU. And she conducted postdoc research at Harvard for 5 years, where she was awarded the very distinguished and prestigious Ellison Fellowship. And during this period, in addition to her neuroscience studies, which we all know are very simple, she was able to have 2 children and raise a family while doing her postdoc. She's been working in molecular biology and brain-related diseases since 2001. Today, she is a principal investigator in the Department of Life Sciences at Ben-Gurion University of the Negev, where she opened her own lab whose research focuses on DNA damage as a cause of aging and age-related diseases. Debbie, the computer is all yours. A machshev shalach.
Thank you. So thank you very much for the invitation, and I hope everybody is happy there in Canada. I will tell you about why do we age and the relation to DNA damage. So we know that aging is a global concern as the population is getting older. And it's a global concern because as we age, we have a more risk to develop many of the diseases that we know, like immune system decline, metabolic syndromes, Alzheimer and other neurodegenerative diseases, heart disease, cancer, and more. So why as we age, we have more disease, and why do we age? And for this, we have to understand very basic concept of how an organism is made. So each one of our bodies is made of cells, and these cells have the DNA that has the code for all the things that we know how to do, our brains, our stomach, our skin, and this is tightly packed in chromatin. If we could take the DNA out of each one of our cells, it will be 3 meters long. So the cell really needs to compact it, but it also needs to leave some regions open and some closed to be able to read the message. So this opening and closing of our DNA is called the epigenetic code. The epigenetic code are like little flags telling I am an important gene in here, use me or don't use me, you don't need me in here. And this is like a script. If we can imagine that the genetic code is a script that every actor, every cell in our body gets, then every actor, if I am a skin cell or a brain cell, I have to say different things. So every actor can mark in the script what it needs to be said. And this allows us from the same genetic code in all of our body to get different cell types. And this gives the identity of the cell, but the epigenetic code is also flexible and can change as we age. And one of the things that many researchers notice is that as we age, our epigenetic code changes, it gets modified, and not all our organs age at the same rate. And this could be related to genetic factors, but also to environmental factors. If we look, for example, at this lady here, you can see that her average age is 44, but her breast is 67 in terms of epigenetic modifications, while maybe her ovaries are 31. So why does the epigenetic code changes as we age? And to understand that, we have to understand that one of the main theories of aging states that we accumulate DNA damage, and this is one of the driving forces of aging. So where is this damage coming from? Every cell in our body gets damaged because we are exposed to chemicals. Sometimes it's even harsher when we have chemotherapy or other kind of treatments. From sunlight, we get damaged. But also just by using our DNA to open and close to read a sequence, we can damage it, or we produce metabolites that can damage it. So the statistics are very different, like skin cells can get 1 million lesions per day, while other cells may get 1,000. But our biological system tries to repair it, but biological systems are not perfect, and we tend to accumulate damage. And of course, as we live longer, we accumulate more damage. So if we have DNA damage and everything goes right, the damage is repaired and our cells are okay. If we get too much damage that our cells cannot cope with it, our cells can be altruistic and commit suicide. If a cell senses that it's too damaged to continue and may become dangerous, it can suicide. But then we lose tissue. For example, skin or in the brain, it shrinks as we age. On the other side, if we don't signal that this cell is damaged and it keeps dividing, we get cancer. And in the middle, we have a state that is called cellular senescence, that the cells are not completely healthy. They are doing half job, but still there. But they are all the time calling the immune system to say, hey, watch me because I'm not completely okay. And this can produce inflammation. So how this affects the epigenetic code? Imagine that every time that we break the script and we have to correct it, we might not do it perfectly. So we may not put all the epigenetic modifications back in place. So we also can lose genetic material, lose important information, but also we cannot put it all back together. And this accumulates as we age. So how can we study aging in the lab and not waiting 100 years to get some results? And for this, we use a gene that is known to be involved in the aging process. This gene is called CIR6, and of course it's not important that you remember the name of the gene, but it is an important gene in DNA repair. When the DNA gets broken, it helps fixing and putting it back together. But it's also important to put these flags of the epigenetic code to signal, I am an important gene, use me, I'm not important, don't use me here. So what happens when we delete this gene, we take it out from our mouse model? These mice present a premature aging and progeria. They look like really, really old mice when they are only a few weeks old. So this presents a pathological aging, a very accelerated aging. On the other hand, if this protein is overexpressed, we put another copy of it, mice can live longer. So it is telling us that this protein is really, really important in the aging process. So what happens when we don't have it and when we have DNA damage? We begin a cascade of events. We push the first tile that will affect all of the processes in our body. So how is this relevant in our body? And for this, we focus on the brain. Our brain weighs only about 2% of our body weight, but uses 20% of the oxygen and 25% of the glucose of the body. So it also generates a lot of toxic waste. Another thing that is important is that neurons, they don't divide. We have to keep them for a lifetime and error-free. So it's more difficult if the cells divide, we can get rid of one that is deficient and the other one stays with us. But neurons, they have all these connections and all this information that we don't want to lose them. So they have to keep the genomic stability for 120 years, hopefully. One of the hints that we get about the importance of DNA repairing the brain is all these mutations in repair proteins. When we don't have them, when someone is born with a mutation in these proteins, they have microcephaly and neurodegeneration and epilepsy and many neurological and neurodegenerative syndromes. Also, in aging patients, we have more damage, but even more in patients with neurodegenerative diseases such as ALS, Parkinson, or Alzheimer's disease. And I just want to comment that 95% of the cases are sporadic, at least for Alzheimer's disease, is not familiar mutations. We don't know the case, we just know that they occur as we age. And in Parkinson's disease, for example, is 90%. So most of the reason of why someone will get Alzheimer's and someone won't is not genetic, it's sporadic. Just 5% of the cases are really genetic. So we try to understand the aging process in the brain, and for this we generate mice model that don't have this protein CIR6 only in the brain. And what we see is if we compare our gene expression, all the genes that are being used of our model with data sets from all of the things that you can imagine, from cancer, any disease, anything that you can compare, the top categories that we get is Huntington disease, Alzheimer's disease, oxidative damage, and Parkinson's disease. So we know that our model is a really good model to study neurodegeneration. We also did behavioral tests and we see memory and learning impairment, and we also see the signs that are known to happen in neurodegenerative brains, like an Alzheimer's disease, start appearing in our mice model. Is this relevant for patients? Yes. When we look at the levels of CIR6 in an Alzheimer patient, it is really, really reduced, and it correlates with the, how harsh is the disease. If we are more impaired, we have lost most of our memory, we will have even less CIR6. So our message is that when we are young and we have normal repair capacity, our brains are healthy. But if we get old and start losing this expression of this protein, we start developing learning impairments and start having toxic aggregates in the brain and sleep problems and metabolic problems that lead to cognitive decline and neurodegeneration. So now what we try to do in the lab is to understand if we can stop this domino effect and also to be able to intervene at different time points. So we see that there are many, many things happening in neurodegeneration, such as DNA damage and cell death and toxic aggregates and sleep problems and metabolic problems. And what we do is we understand, first of all, what are the biological causes of aging and neurodegeneration. This helps us to find pathways that can be targeted. We have some molecules and some pathways that we are already targeting and trying to improve the cognitive capacity of our models. Then we can also develop diagnostic tools to find earlier who may develop the disease and also test compounds. And our objective is to get to clinical trials and to cure and prevent disease. So this is part of, from basic science, how can we get to the big picture and try to help people that are aging and may develop a neurodegenerative disease. And this work has been done with the help of all my wonderful students in the lab. Monica Inav, Katerina Ermenko, Shai Kaluski, Miguel, Daniel, all of them are really, really amazing students, and our collaborators all over the world. And that's it. I think I made it on time. So Mark, I'm back to you.
Whoa, hold on.
Okay.
Unmute, start video. Here I am. Tammy, Debbie, you came in under the wire. Unbelievable.
I'm afraid I wasn't finished.
No, it was terrific. You spoke quickly, but terrific. I understood this much. Simon, questions, please.
Yes, thank you, Mark. There's a first question that we received actually before the beginning of the webinar about early detection in neurodegenerative diseases. Can you comment on that, please?
Tammy, do you want to begin?
Maybe you will start and I will.
Okay. So what we are trying to do, or what we have been able to see, is that there are some categories that we can compare aging to pathological aging. And we have certain genes that are always expressed in aging, but not in pathological aging. When we compare with Parkinson's disease patients or Alzheimer's disease patients versus just regular people, we see some categories that are changed. And we hope this will help us develop diagnostic tools. The problem is that we analyze brain samples, but you cannot go and get a brain sample from a patient. We try to translate our findings to things that can be found in blood tests. And this will make things more simpler. But we are not there yet because not every change that we see in the brain is also happening in the blood. And maybe Tammy technology could have a better, without intrusive technology to solve this.
Okay, so I think we addressed this question in a different manner than Debbie, and this is good. So what we are aiming to do is to train a neural network, and the hope is that such neural network will be so accurate. Now, the assumption is that if brain age and chronological age are identical, then the subject is healthy and everything is okay. While when we do overestimation of brain age, that it is much older than chronological age, then there is some issue or suspect that the patient may be suffering some neurological disease, and this is our hope. But we still actually do that.
Okay, thank you. There's a question about what is known about the hereditary component of Alzheimer's?
So most of the mutations known for Alzheimer's disease are mutations in a protein called amyloid beta or tau. These two proteins, when mutated, they cause the disease very early. It can be in the late 20s to 40-year-old patients. And one of the things that is known that amyloid beta accumulates in the brain, and it was thought to be the main marker of Alzheimer's disease. But now it's known that you can have a brain full of amyloid beta and be completely healthy, and it has no correlation with the severity of the disease. Tau, on the other hand, is a marker for more than 17 neurodegenerative diseases, also Parkinson's disease, and it's also present in Huntington disease and in many, many other neurodegenerative diseases. And its accumulation, it is really correlated with the cognitive decline. But these mutations occur only in 3 to 5% of the population. So 95% of the population that get Alzheimer's don't have these mutations. So the cause has to be something that goes across all the different places on Earth and is age-related, because the main risk factor for this disease is aging.
Great, thank you. There's a question about do cognitive decline and dementia manifest themselves the same way in patients with Alzheimer's disease and those with Parkinson's disease?
Tammy, you want to say something?
Well,
No, okay.
I'm not sure. I'm not sure if I can answer this question.
Okay,
Yeah.
so in Parkinson's disease, the most characteristic thing is trembling. They have a complete, at the beginning of the disease, the memory is not impaired, but the first symptoms are more trembling and loss of control of the body movements. In Alzheimer's disease, the first thing that goes, that gets lost is memory of new memories. Some of the patients can conserve old memories from childhood, but start with decreased capacity to remember novel events. So these are two different characteristics that in the end, as the brain declines, also in Alzheimer's patients, they may lose part of the body functions, and also in Parkinson's disease, they start also getting impaired memory.
Thank you. There's an online question which is quite detailed, but I will read it out to you, and I feel it's quite important as well. Mr. Bernard Katz, my wife has idiopathic normal pressure hydrocephalus and was shunted in April 2007. Over the past few years, her shunt has become less effective, and after a battery of neuropsychological tests, her very experienced neurologist and her neurosurgeon have proposed that advancing Alzheimer's has somehow started to interfere with the shunt's functioning. The shunt itself has been proven to be functioning. How can we determine whether someone actually has Alzheimer's?
I don't know about the shunt and the other phenotypes, but Alzheimer's disease is mainly diagnosed by the kind of memory loss that is happening with cognitive tests, but also there are some novel technologies that can measure this tau protein that I was talking about and its accumulation in the brain in a non-invasive way. So these are the two main ways to test someone. The less invasive and less expensive one may be the test of the type of the cognitive decline.
Thank you. There's a question from Dr. Keith Meloff. What is the role of sarcopenia as a contributor to dementia in the elderly?
I really don't know.
Okay, not your field. The question from Mr. Gad Albaz in Montreal: how much truth is there in the assertion that activities that engage the brain, such as reading, learning, crossword puzzles, musical instruments, or a new language, are effective in delaying or preventing certain diseases?
So from what I know, there is no scientific data, like real well-developed studies that prove that this has any effect. There are many mails and going around Facebook and WhatsApp and whatever telling that you should use your left hand if you're right-handed, or if you should learn a language. All of these things will be great because it keeps you alive and happy, and depression does correlate with a higher incidence of neurodegeneration. But no one has really proven in a scientific and methodological manner that all of these things really prevent neurodegeneration.
Thank you. So we could still do crossword puzzles just for the pleasure of it.
Exactly. No, maybe they work. I am not saying that maybe it doesn't work, but there is no one that has done a really methodological comparison that can give us certainty that this works.
Okay.
It's
Thank you.
just like a common knowledge going around, and no one has really tested it properly.
Okay.
Sam, could I just, Debbie, when you were out west, you did mention that two or three items in one's life might contribute positively to the slowing down of Alzheimer's. And I remember one was oxygenation of the blood, exercise. One was turmeric. And I can't remember the third one.
Yeah, the turmeric I don't remember, but diet can contribute in a bad way. Like calorie restriction has been shown to prevent cognitive decline. I don't know if really prevent if you already have Alzheimer's disease. So this is one of the things that decrease aging, sport, and the things that high-fat diet or unhealthy diet can make it worse. Metabolic syndrome is one of the second risk factor after aging. Metabolic problems can contribute to development of Alzheimer's disease, and depression and schizophrenia and other diseases can also increase the chances of developing Alzheimer's disease.
Yes, I remember you had said the 16-8 diet was found to be perhaps effective.
Yes, at least in animal models, and many people are trying this nowadays. So maybe we'll have some answers also about this. But it has been proven in people to reduce metabolic syndrome. So if we can reduce metabolic syndrome, also our chances of developing Alzheimer decrease.
Thank you. We have a couple of more questions. Regarding identifying low levels of SIRT6, is this something that can be checked earlier in one's life in order to address it earlier? And the second part of the question is in terms of medication or pharmaceutical medication for Alzheimer's disease.
So regarding SIRT6, we see that it declines as we age. Also in blood samples, also in the brain. But there is like a catch, because some tissues really require high expression of SIRT6, like the brain, to prevent neurodegeneration. But some other tissue, if you have too much of it, may cause cancer because it's protecting cells from dying, and maybe these cells should die. So what we are trying to do is to increase the levels only in the brain, because we don't want this duality of this protein. And the main question is if in the blood can reflect what is happening in the brain, and it seems this is the case, but we are not completely sure. And regarding drug targets, we are working on some at the animal model, but it will take many years to get into the clinic, because we have to make sure they are safe and they have a good effect. But the good thing that I believe is that for more than 30 years, the only thing that has been targeted is the genetic causes, and they cannot work for the other 95% of the population. By targeting aging and age-related diseases, I think that we will get more promising drugs for most of the population.
Thank you. There's a question about, are you familiar with the work of Dale Brendeson's The End of Alzheimer's? It has been recommended as reading for one of our people. And they're wondering if you're familiar with his protocol, and they were told that it's helpful to find out if we have one of the APOE genes. Can you specify and comment, please?
Which gene?
The APOE, as per the
Yes,
question.
okay. So one of the only genetic factors, genetic markers that has been proven to increase the chances of getting Alzheimer's disease is APOE4 gene, that it's related to lipid metabolism. And if you have this variant, you have increased chances of getting Alzheimer's disease, while if you have the variant 2, you have decreased chances of getting the disease, but the increase and decrease is not like 100% chances. This is one of the genetic factors that is not causing the disease properly, but increases the chances of getting the disease. And regarding these studies from this scientist, I don't know, but I will check. If you write me the name, I'll be happy to check. There are, I guess, thousands of people working, or millions of people around the world working on Alzheimer's disease. Yeah. And everybody takes a different aspect of the disease, but I'll be happy to learn.
Thank you. We'll send it over to you. Yes. Question, what would be the effect on brain aging from spinal stenosis and surgeries that relieve pain from that, such as laminotomies?
I don't know.
Yeah.
Tammy?
No.
No. Okay,
no.
Let's move on. We have a question. Is it important to emphasize that Down syndrome is a genetically determined disorder that leads to Alzheimer's disease? Is there a connection?
Okay. So in Down syndrome, the extra chromosome is also encoding for one of these genetic genes that can lead to familial Alzheimer's disease. And that is why many Down syndrome patients can get Alzheimer's disease or neurodegeneration. So it will fall into the category of familiar cases.
Great, thank you. I think that we have completed our questions. Mark, I'll bring it over to you.
Simon, actually, I just got one. Someone sent me an email wanting to know from Tammy what reintroduced you and why are you so interested and fascinated by the brain science, by the science of brain?
Okay. Well, it's a long story, but well, I'm interested in the brain for many years. Somehow, so actually, I started with a great interest with the brain. I moved on to computer vision because I thought that it will help me to understand, things about human vision throughout the engineering and computer science model of visions. But then I found out that I'm doing the other way around. That is, I'm exploring computer vision and machine learning tools in order to understand the brain, and this is how I get back to it.
Okay, and I see there's also another question that just came online, and I think this is because of everyone who is so drawn to this keto diet. This is from Steve Lewis. I have adopted Dr. Bredeson's reading on using ketones instead of glucose for fuel. After 2 years on keto T2, after 2 years on keto, T2 is reversed and weight is low and stable. How well researched has the neuroeffectiveness of a ketogenic regimen been clarified, if at all?
So
Last
there are
question.
many research on animal models, and again, I can say many things about rat and animal models, but I know less about people clinical trials that show that for many neurodegenerative or neurodevelopmental disease, they see a huge improvement using ketogenic diet. So one of the models of cocaine syndrome, for example, they have kind of neurodegeneration, and they have like many age-related phenotype and accelerated aging. And they show by switching to ketogenic diet and also reducing the calorie amount, but not too much, that these mice live longer and were doing much, much better in their cognitive and physical tests. So at least for animal models, it has been shown to have a very good effect, but it has to be considered that it's not good for everyone, the ketogenic diet, and it may have some everyone will have to adapt it. If you have heart disease or problems with cholesterol or other kind of things, it should be considered, and not everyone should just jump to that. But my knowledge is in animal models mainly.
Okay, and my love is eating the animals when they're cooked. Once again, thank you, toda rabba, to our panelists. Another fascinating webinar. I think that, and all of a sudden we started getting at the end, about 10 questions popped up, and I've gotten about 4 on my phone. So what we're going to do is write them to you guys, and perhaps you would be able to find the time in your busy schedules to give some answers. I want to say a toda rabba to my excellent team across Canada who have told me today not to mention their names. They said it. My national president, Mitchell Olbaum, a great mentor, advisor, and friend, and his leadership team. Thanks as well to my excellent colleagues in Israel, President of Ben-Gurion University, Danny Shamovitz, Vice President Jeff Kay and his team, and Doug Sesserman, CEO of the American Associates and his team. Please check us out at www.bengurion.ca. I now have a couple of very important announcements. I am taking this opportunity to officially launch our worldwide SOS Support Our Students, Save the Class of COVID-19 emergency campaign. Unfortunately, the COVID-19 situation has literally devastated the economy in Israel, and more than 1 million people, over 25% of its workforce, are out of work. And that most definitely includes the university students and its population. BGU finds itself in a crisis situation unlike any other it has experienced in its 50-year history, and estimates that there will be between 2,500 and 3,000 students desperately seeking assistance for their tuition, room, and board. President Danny Shamovitz has appealed to all the associates offices around the world to come together and has asked that each of us launch an emergency campaign whose total goal is now guesstimated to be between 5 and 7 million US. We in Canada will be approaching our support base, who will be asked, and I quote BGU's president and vice president, to give until it just might hurt. Our first e-blast about it actually went out last night at 8 p.m., and I personally have responded to the president's request and have given till it hurts. I even got my wife commented on it, but she's very happy that I did it. This is truly a situation of unprecedented consequences, because if we do not have students in our university, we do not have a future for not only the Negev region of Israel, but for Israel itself and the entire country, and it's being known as a startup nation. Now that's on the downside, and I have to tell you that to date, I just got an email from the university that about 2.5 million has already been raised and pledged. So we are well on the way, and people are giving and answering the call. I'm also very proud to announce that on September 9th, we will be holding a national virtual gala event featuring Mayim Bialik, a neuroscientist, but much more known as Amy Farrah Fowler of Big Bang Theory. President Danny Shamovitz will also be presenting that evening, and all proceeds will be going towards the Support Our Students emergency campaign. We have some goodies in store for everyone who purchases either a sponsorship or tickets. You will receive an outstanding bottle of red wine from one of Israel's top vineyards, Yatir, along with 2 engraved wine glasses with the logo of the university and a box of eating treats. And depending on where you live in Canada, they'll either be sweet or savory. Since we're having it at 7:30, it's 7:30 in the East Coast, but it's 4:30 in the West Coast. And here is a special short message from Mayim Bialik herself.
Hey, this is Mayim Bialik. You may know me as Amy Farrah Fowler from The Big Bang Theory, or maybe you know me as Blossom. I'm going to be taking part in the Canadian Associates of Ben-Gurion University's Virtual National Gala, September 9th, 7:30 Eastern Standard Time. It's taking place all across Canada. It is taking place in Montreal, Toronto, Winnipeg, Vancouver, Ottawa, and the Atlantic provinces, which I had to look up because I didn't know what they are because I'm from the United States. All the funds raised will go to the Ben-Gurion University SOS. That's the Support Our Students Emergency Fund, which is helping those affected by COVID to be able to continue their important studies. So please join me. It's going to be really fun, September 9th. Hope to see you there.
Thank you, Mayim. She was terrific on. I think she really complimented the show in such a tremendous way. Big Bang Theory. Our 10th webinar, which also might be the last before the summer, will take place on July 29th, featuring students from Ben-Gurion University, each of whom has been affected by the COVID-19 pandemic. Thank God, though, not in a medical way, or they haven't been sick, but they have been affected financially, emotionally, and organizationally within the university. These are among the best student leaders at BGU, and are certainly within the group that we love to call the best and the brightest. Join us as Jeff Kay, Vice President, will be leading what should be a fascinating and personal insight and discussion with we had a talk with them yesterday, and their experience over the past few months is very touching, and they will be able to explain the life of a student during these terrible times when all of a sudden classrooms are canceled, their part-time jobs are canceled, and they do not have money, neither from their parents nor themselves, to be able to pay for tuition. I wish you all stay safe, wear a mask, social distancing. I mean, the three non-scientific or scientific but non-neuroscientific easy steps to follow: mask, social distancing, and that's it. Stay safe. See you next week. Mark out. Thank you, everyone, for attending and participating.